just let me know when you want.
All right, first thing is I put up some signs this morning. There are a couple of research seminars coming up about biotechnology, and these are scientists from the Root Institute who are working at USM and are our colleagues. And we're working together to create a bunch of more research opportunities for students, and that's going great. Okay, so like I said, I put signs up. And then I apologize, I haven't finished grading your exams. I grade the exams one question at a time, so I haven't finished grading anyone's exams yet. I pride myself in getting exams back the next class period. This is the next class period. I had a weekend to grade them. I didn't get it done. I might not get them done by Thursday because I have a lot of other stuff going on. But I'm doing the best I can, as I'm sure you are as well. Now we're done with the second exam, and we have a little bit of new material for our final, which I didn't put up here, but I think it's 2 weeks from today. So when 3 weeks from today you like to think, who likes to think differently than that? You think we have 3 more weeks of classes? What do you think, Lydia? Lydia does not know. I heard her mouthing, I don't know. So that's it. We don't know. But it's coming out. And when we were studying for the second exam, we were talking a little bit about studying for the final, because the final is the same style. It's just questions I didn't put on the first 2 exams, literally. You know, you're expected to just be able to go back and loop through everything, and there'll be some new material like that, and but it'll be mixed in, and you'll see we just keep stacking stuff up. So what are we going to read? We're going to read one more chapter. It's called The Nucleus, and and it's a good chapter for us to go back to earlier in our textbook, right? So for our second exam, we finished the end of our textbook, and it's good to go back into the core of our textbook because now we have plenty of examples that will be satisfying to see when we learn something about the nucleus like that, right? We spend a lot of time talking about stuff inside the nucleus, and so that's good. And then beyond that, we'll just keep talking about new material, and but you won't have additional reading. You also will not have any additional assignments except for the term paper. So that's the remaining of our class, is keep coming to class, get ready for the final, and work on your term paper. And so your term paper work has these really 4 parts to it. I shared 2 documents with you. Then I described the term paper in the syllabus, and then in these 2 videos that are at these different Brightspace announcement places, right? So the one is the original core of what do you do, and then the second one recently I just did an update to answer questions that were general knowledge questions, you know, that people were asking about that we're working on their term paper. And so let me know if you have questions, and you can bring them to class like that, because if you have questions, we do. The official due date, believe it or not, was this Thursday. If you can get it done by then, great. And the way you get it done is you just are done working on it, right? Like that. It would be a good idea if you could send me an email and say, I'm done with my term paper, so I know I can grade it. Otherwise, I'll be just not worrying about you because I think you're still working on it. Even beyond the end of the semester, if you don't let me know you're done, I'll just be like, how would I know, right? Yeah, so you know that's a feature that I haven't mentioned before, and that is I need to know when you're finished with the documents like that. And so that's great. And you don't need to send them to me. I've got access to them. You know, you can just let me know. And you can let me know in person, or you know, in class you could say, hey. So that's that. So we're working on that. All right, that's where we're at. And let's just look at this. Here's our textbook, and let's just reflect on our textbook is made up of 19 chapters, and you will have read at the end of this semester chapter 1, chapter 7, 16, 17, 18, 19, and 10. Okay, and so remember, I'm like, the book has too much material for us to learn. So we're gonna, you know, work our way through it. Remember the first time I taught this course, there was a textbook that was like this. And I just said, I think that textbook is really well written. And I would just answer questions as they come up. And we would just roll through big chunks of that textbook. And that's true at a lot of schools. Of course, a lot of schools use this, a lot of different instructors, and a lot of instructors are just, take a look at the book, you know, expensive book, you know, that, and just roll through the chapters one after another, you know, like that. So one of the things that, again, I really work on is trying to talk about how to learn biology. Here we are at an advanced course. A lot of you are graduating in 3 weeks or however long it is, you know, and yet, you know, still talking and thinking about learning about biology. I am still talking and thinking and learning about biology, right? I think that's really an important part of, you know, this experience, is you're not just picking a book up on yourself and by yourself and trying to navigate it through. So this is the structure of our course, our textbook. And so we took some of the intro stuff, and it introduces these really 2 topics that our book, our course is. It's called Cellular and Molecular Biology. It really means 2 topics, cell biology and molecular biology, that of course are really overlapping, and nobody worries about that distinction, but they are distinct fields. And so this second section is really a lot of introduction of molecular biology. This final is a lot of application, you know, in a specific example. It's this third section that's really the cell biology section. So we're just going to deal with 1 chapter of cell biology, and it's about the nucleus. So of course the nucleus is an organelle. Nobody knew it existed until, you know, about the 1940s or so. And it really became prominent as well as other organelles when people could see them under an electron microscope. And so our book has pictures of it. And you'll see the topics in this chapter have this more holistic story to them, where you're like, we can't make a transgene, you know, it's more of a bigger picture where we need to have these parts fit together. And in general, students really like cell biology. It's a satisfying thing, because you have to kind of see it all put together to understand how it works. And you know, we're moving in different locations. We're inside the nucleus, we're outside the nucleus in the cytoplasm. So, you know, in a sense, you know, right, our author was intentionally, we're going to introduce it, then we're going to stack up some molecular biology, then we're going to stack up some cellular biology, then we're going to do some cancer biology there, right? Put them all together, right, like that. So that's our flow. And the chapter is made up of these 3 parts. The second and third part are really light. You know, there's detail, but they're really light. The concepts that we want to get out of them are just like 10, 15 minutes of talk, like that. Maybe there'll be an extra 2-point question on the final exam from those parts, like that, but they're not huge. The first one is really satisfying, and it's about the membrane around the nucleus. So the nucleus is surrounded by a nuclear membrane, and that's what it's about. And a big part of it is that here's the nuclear membrane, and this is a gate, a door in the nuclear membrane called a nuclear pore. And things have to get in and out of the nucleus through the nuclear pore. And it is a gate. It's a regulated gate. So we haven't talked about that before. We've talked about stuff just going in and out of the nucleus. But there is a regulated gate in the nucleus, like that. And so we'll talk about that. To get into the nucleus, a protein needs something called a nuclear localization signal, and it's just, or it's abbreviated NLS, and it's just a very short part of the protein. And so we can just draw one right now and say it looks like this. So let's just say we talked about heat shock transcription factor, HSF, heat shock transcription factor. And all transcription factors are proteins. So that must mean they're made by coding genes, right? And the heat shock transcription factor we have, of course, is the human heat shock transcription factor, as are all the proteins we have in our cells. They're all human proteins, and they had to be made by the genes, and the genes are inside the nucleus. And so if we just look at that story, you know, here's the nucleus. And here's the gene for HSF. And we're using this as an example of a transcription factor, and in our body there are about 2,000. And we learned several of those, right? So this is just generally true about a transcription factor, is it's a protein. And it's got to have a gene. And the gene is a coding gene. So there it is, and there is the coding region. And there is going to be a cis-regulatory region, and it's going to be activating this. And we could ask, well, what cells is this activating? Is this gene activating? And the answer is all of them, all the time. So every one of your cells has HSF right now, so it can have the heat shock response if needed, and it's expressing heat shock protein all the time. And so constantly in every one of our cells, we could detect, if we did an experiment, we'd be able to detect the RNA that's being made. The gene is being transcribed by RNA polymerase and producing RNA. And RNA and proteins in general are kind of unstable. So if we say a protein is in a cell, this cell is almost certainly constantly transcribing the gene and making the RNA and making the protein. And so one way to study gene expression is just study the RNA like that. Yep. And so it's making RNA, and then when it leaves the nucleus, it's ready to be used by the ribosome. So it's called mRNA, messenger RNA, because it's bringing a message to the ribosome. And the ribosomes are in the cytoplasm. And the ribosome is going to transcribe this into HSF. So we got some HSF protein, right? And if we zoom in on that, we know what it looks like. It looks like this, and I don't know how to draw this, but it kind of looks like this, where it's kind of folded up, right? And we said before, there's a trimerization domain that's hidden, blocked. So right now, all of our cells have HSF, and it's inactive because it's a monomer. And then when there's a little heat stress that could denature proteins, it kind of changes the folding heat shock transcription factor a little bit, and it opens it up and exposes this trimerization domain, and now we get the heat shock trimer. Something we didn't mention before, and that is that HSF also has an NLS, a nuclear localization signal. And that's also covered up. When I described it before, I said, you know, heat shock transcription factor, it's very weak. So if it could try to bind to the enhancer of heat shock genes, it would bind really weakly because it just has one hand to grab hold of. Well, it's not even in the nucleus yet. So part of heat shock activation is to unfold the heat shock transcription factor, expose the trimerization domain, and also expose these NLS. And now, the heat shock transcription factor can go into the nucleus because it needs a nuclear localization signal to get through the door. So we kind of have a double regulation of that protein, right? When it's a monomer, it wouldn't bind very well, but when it's a monomer, it can't even get in the nucleus to try, you know? Yeah? And so that nuclear localization signal is a key thing. And it's just a little portion of the primary amino acid sequence. Our book has some examples of it. I think I can find one right here in a slide. Maybe I have to scroll forward a little bit. Yeah, we'll get to it because we will just introduce this concept, and then we'll talk more about it. So a key concept is that genes are in the nucleus, and any coding gene has to have the mRNA leave the nucleus because the ribosomes are in the cytoplasm. And then the translation occurs in the cytoplasm, and then any protein that functions in the nucleus has to get back in. And to get back in, it needs a nuclear localization signal, and those are just built into the structure of the protein. The sequence of the protein has that little patch, that's a little tag that says you can go into the nucleus. But sometimes it's covered up. And that's part of the regulation, right? You can't get the protein to function until it can get a nucleus. So let's just think here and see if people can suggest some examples of proteins that we've learned about that would have an LS.
FOXO.
What?
FOXO.
So FOXO. And so let's look at FOXO. I have a slide of FOXO here. We started our course with it. There's FOXO. Now we know what is happening to FOXO. When there's stimulation of growth and the environment is good, and let's roll forward and grow up and reproduce, FOXO gets sequestered in the cytoplasm. How does that happen? And we learned, well, AKT phosphorylates it, and then 14-3-3 grabs it and holds on to it. What's happening there? the phosphonuclear localization signal is blocked by 1433. So that's what's happening there. It's just physically blocking it, so it can't get into the nucleus. Yep. Yep, so that's an example. Another reason to think that FOXO would have a nuclear localization signal is not just its regulation of getting to the nucleus, but it's a transcription factor. So when 1433 lets go of it, that nuclear localization signal needs to be there, exposed, and allowed to go in, because that's where it does its job. And it gets to do its job when the conditions are not great, and you should slow down and the organism should try to extend its lifespan and hope for better environments. Yep, so that's one. Yes, more.
Beta-catenin.
Beta-catenin, yep. And we could generalize that to say all transcription factors, literally every transcription factor has to have a nuclear localization signal, because they're all proteins, so they all must get synthesized by ribosomes in the cytoplasm, and then they need to get back in. And sometimes that's regulated, lots of times it's not. They just get made and they go back in. Yeah? But it does provide a good point to regulate them, because if they can't get in, they're not active. Do you have a question, Ken?
Yeah, but wouldn't there be transcription factors that get encoded in the nucleus so they don't need to get entry inside?
No, because there are no ribosomes in the nucleus. Yep, yep. So that's a key concept right there, right? And it's a good one, right? And this is some good cell biology, right? It's satisfying, right? Because you're like, yeah, okay. They couldn't get made in the nucleus, they get made in the cytoplasm, they get made, they have to get back in. So they need a nuclear localization signal. What are more?
Can
Proteins.
we talk about ribosomes? So I know ribosomes are made up of proteins. Is there like a ribosome gene?
Let's talk about ribosomes, but let's wait till
Okay.
later in this chapter, because ribosomes themselves are made up of proteins plus an RNA called ribosomal RNA. And so there's a story there that the book gets into, because to build a ribosome, what happens is the proteins are genes, ribosomal protein genes. They get transcribed, they're coding genes, they get expressed as RNA. The RNA leaves the nucleus. The ribosomal proteins are synthesized in the cytoplasm. Ribosomes themselves are assembled in the nucleus where the ribosomal RNA genes are. So we get a really kind of advanced story of the creation of ribosomes. And we'll just hint at that now, but it's one of the things our book wants to talk about toward the end of this chapter. Because it's just like, whoa, what we took for granted all the way through our course is just, gonna make this protein and use it, right? So yeah, literally every transcription factor and all these ribosomal proteins have to get into the nucleus, yeah? So they all have this nuclear localization signal, which again, is just a really little patch. It's really nothing special. It'd be very easy for a protein to have it. Can anybody think of any other proteins that have to have nuclear localization signals? Sam, did you have a question?
I can
No.
answer your question, and then I do have a question.
All right,
DNA replication machinery.
gets into the nucleus, yep.
It's in the nucleus.
All those DNA polymerase, RNA polymerase, right? All those, anything that has to get into the nucleus to do its job has to have a nuclear localization signal. Yep.
Is a nuclear localization signal bidirectional? Can you use it to get out of the nucleus?
I don't know, maybe not.
And then do they call it a different word? Because I know for proteins that need to
Get in
go-
and out?
Yeah.
Yep, right, yep. They have to get out using a different strategy, yep. And they have to have a signal to get out. And is it the nuclear localization signal? I don't think so, but I guess I don't know exactly what that is to get out. Matt?
Is it just proteins that have to have this coding to get in? Because I was thinking think of like a retrovirus getting in, and that just goes right in, or is that like a different process?
Yeah, so that's a good question, and the answer is that, say a retrovirus, when it comes in and infects a cell, right, it's going to copy the RNA genome, transcribe it using that reverse transcriptase that we talked about, and then it's got to get that DNA into the nucleus, because it's got to insert into the chromosome, into the DNA of the host, and get transcribed there. And we'll see in this chapter that RNAs and DNAs that are in the cell, they're always bound by proteins. And it's really the proteins that are the thing that has to get in and out of the nucleus. but because of that, the entry into the nucleus, the DNA for the virus, that would also be regulated. So it's gonna have to have some proteins bind to it to allow it to get into the nucleus. Yep. So it's just a surprise, because up until now, we're just like goes in, goes out. But it's a gate, and it's regulated. And it's regulated in, at least at the beginning, a seemingly simple way, like we just learned about heat check transcription factor. You have to have the nuclear localization signal, and then it has to be exposed somehow to get into the nucleus. Yep.
Does it use ATP to go in? Is it like active transport?
Or GTP, but we'll talk about that. Yep. Is it an energy dependent process? Yes. And we'll talk a little bit about that. Other proteins that would need to get nuclear localization signal get into the nucleus. So we talked about histones. So structural components, proteins that are structural components of the chromosomes, they have to have a nuclear localization signal. Yep, lots and lots of stuff. RNA polymerase also has to have a nuclear localization signal. Yep, lots and lots of proteins have to have it. Just a huge amount of traffic going in and out of these gates. Yep. All right, do people have questions about that? Okay, so then, so then we're gonna pause because we've been taking it light on this topic about the idea of, well, you gotta regulate that, right? You know, we really didn't talk in detail about how that FOXO really can't get in, you know? It gets phosphorylated, it gets bound by 1433. So what does that mean? And the answer is its nuclear localization signal is covered up. Then we mentioned the trimerization of heat check transcription factor, but there's also the localization signal, nuclear localization signal as part of it. So we have to do something now, and that is we want to talk a little bit more about the pathway of gene expression and how any of those steps could be regulated and affect the activity. So the way to think about this is to start out and say, here's a gene, and we've got all the genes that we inherit from our parents. So a big part of the story of life on Earth is regulating gene expression. Because that's not just true of humans, that's true of every cell on our planet. Even the simplest bacterial cells have more DNA, more genetic information than they're using at any given time. So this regulation of gene expression is hugely important. Regulation of it, turning it on and off in a controlled way. And we've really focused on transcription regulation. That's the very first step, and it's very often regulated. So we're gonna get it translated, transcribed, you know, and that's called transcriptional regulation. And it's all about transcription factors. And we're just gonna pause for a minute and say, well, that's not the end of the story. We mentioned this a little bit with MYC. You know, well, you know, the RNA polymerase has a hard time transcribing MYC. Then the RNA for MYC is very unstable. Then the ribosomes don't want to translate it. Then the protein's unstable, you know? So those are all examples of how to regulate gene expression after transcription. So we can talk about transcriptional regulation gene expression, and we can talk about post-transcriptional regulation of gene expression. And we could have added that at any point in our course. It kind of adds complexity, you know? We have added it in lots of different points. For example, we talked about just now FOXO. That's a kind of post-transcriptional regulation, right? You can make the FOXO protein, but it's not active. So it's not like you even made it, you know? So that's really important, heat check transcription factor. So even though we didn't talk about the broader concept of post-transcriptional regulation, we've talked about many examples. So we want to just look at this and talk about this a little bit, because that's where the nuclear membrane comes into play. That's a step for this post-transcriptional regulation. So this is just from an intro textbook, and it just shows the pathway. You know, here's the DNA. We're going to transcribe it. Transcribe it into RNA. And a word we haven't used yet is when the RNA gets transcribed, it's called the primary transcript. I don't think we've mentioned that in our course yet. And then, before it leaves the nucleus, it gets processed. And so that's referred to as RNA processing. And we'll talk about some of that toward the end of our course. But that's a place where regulation can occur. Then it leaves the nucleus, okay? That's what we're talking about in this chapter, the ability to get out of the nucleus, that's regulated. Then it's going to get translated, if it's a coding protein, a coding gene, and then the protein is going to be there. And so really the post-transcriptional steps in regulation of gene expression that people like to talk about, because there are a lot of really good biological examples of it, are this step here that's inside the nucleus that happens to the RNA, and that is generally called RNA processing. And then the other one is after the protein is made, it would be post-translational regulation. And we've learned a lot of examples of post-translational regulation. That phosphorylation of FOXO and the binding of 1433, that's post-translational regulation. All the story about activating those CDKs, right? They just get made. CDK1 is always there, but it's being regulated in a post-translational manner, like that, right? Yep.
Is that signal like the start, like it's going through the green light at the very top?
No, the signal is not going through the green light. The green light was cell cycle regulation, where we talked about that. The signal would be like activation of transcription.
Okay.
So the signal would be something like the estrogen hormone could be a signal to activate transcription. Or, it could be more removed from it, like PDGF functions a little bit like activating transcription, right? Because that's the cellular response, is let's activate gene expression and then go through the green light.
Okay,
Yep. Yep. Other questions people have? And we want to be reasonable about this, but again, it's an intro bio concept, and it's a cell biology concept, because you can see there are these physical locations where this regulation is taking place after transcription regulation, after transcription is done. So in this chapter, we're just focusing on the nuclear membrane, and either getting in with a nuclear localization signal or getting out, okay? And so that's what it is. It's about traffic, movement of molecules back and forth. And a lot of small molecules can just get through, and but these larger molecules like proteins, DNA, RNA are regulated. And it's a cell biology topic, because back in the 1950s, when scientists first got to use electron microscopy a lot in biology, they saw, oh, the nucleus is decorated with these little donuts, and the little donuts are called nuclear pores. And there's images in your book about it, and here's just a picture of a nuclear pore. So it's just a donut, and the donut hole is the gate. And it's a really complicated structure. Bunch of proteins are participating in it. And, structurally, at first it seems like it could just be, it's just a hole in the nuclear membrane and stuff can get in and out. And up until now in our course, that's what we were just thinking, just get in and out, Once 1433 lets go of FOXO, it'll just go in, right? Well, it's regulated. And so the first level is this structure, and we're not going to worry too much about the structural details, like all these different components, because the structure is often kind of just seemingly kind of a boring story, like that, We're like, it's a gate. The real key thing is the regulation of the gate, what opens and closes the gate. Yep. So we want to get onto that to talk about it. But at this level, a cell biologist would be like tackling me. If I say, oh, this feature, these structural features, well, they're just pretty boring, pretty static. Yeah, and they would like charge me and knock me down, which would be cool to see, yeah.
So the image of the picture says the nuclear lamina, but then it points just to one yellow thing as if that's a lamina. Is what part would be phosphorylated by like the CDK1 complex?
Yeah, so let's answer that question by saying, the book plays the cell biology role here. So the book says, so you see this structural stuff here, and it says, see that tan stuff? Those are the chromosomes, they're cool and important, but they're just chromosomes, whatever. Then it shows these little yellow things, and those are the nuclear lamins that you're asking about. Those get phosphorylated by CDKs as part of start. Part of AMP, right? Sorry, yeah. So CDK1 cyclin B phosphorylates those, and they disassemble. The nucleus breaks down in a lot of different species. Yep.
So that membrane, like that,
Yeah.
all that goes
Yeah.
away.
And the book actually picks on these nuclear lamins, because you're like, I see them, and I know they gotta get disassembled during mitosis, but this is just during G1 when the cell is just doing its business, right? So the book says, you want to know something curious? So these nuclear lamin proteins, they're proteins, so there must be a nuclear lamin gene, and there's a famous genetic disease, heritable disease, that there's a good chance you've heard of, that is mutation in a nuclear lamin gene, okay? And so the book is just doing this cell biology thing, right? Where it's saying, shut up, Dave. This is not a boring structural story. There's crazy stuff happening in this picture. And here's an example, right? So there is that little drawing out of that nuclear lamins. They're monomers, they get polymerized, and if they get phosphorylated, they depolymerize to the monomers during M phase. So we learned about them. That's what we know about them. This is their role as a boring structural component of the intact nucleus when genes are being expressed and so forth. And there's just a picture where they're located. And here's a kid that's a young kid who has this fast aging disease that's called progeria that you've almost for sure heard of, where a child could be 5 or 6 years old and literally be the biological age of a 90-year-old person. And they have a mutation, at least one category of this disease has a mutation in one of the nuclear lamin genes. The nuclear lamin genes are a gene family, and they have a mutation in a specific nuclear lamin gene. And no one, as far as I know, no one has a clue why, some damage to the nuclear lamina would cause rapid aging in a human, And so you'll see that in the book, right? And the book is doing that with intention, right? It's picking on this, and I don't think it says outright, but it's saying, like, if your professor is a molecular biologist and he starts talking about this cell biology as being pretty static and boring, either tackle him or throw something at him, or just get up and walk out. Yeah? I had a friend named Clark was in a class, and Clark was such a jerk. Remember, I got booed. Clark was an English major, so I had no classes with him, but he was such a jerk in class. One time, one of the professors left. Yeah. He's like, the professor says, it's English, so he says something like, such an ostentatious outburst from such a casual member of our class. And he just left. So you might do that. You might reverse it and just leave and say, cell biology is cool. Yes, Alex.
So the mutation, it's a deletion?
It's a really curious mutation because it's involved in getting the gene expressed, and we're used to thinking, okay, so it might be in the cis-regulatory region. This one is not. Then we say, well, then it's in the transcription unit, and it's a protein, so it must be in the coding region. It is not. It's in the transcription unit, but not in the coding region. And I've shown and drawn that each time, that there's the transcription unit, and then there's not all of that is the coding region. And these kids have a mutation that's actually outside of the coding region, but in the transcription unit. And we just want to be gentle on ourselves with that reality, right? Because we have a lot on our plate to remember for the final, right? The parts, 2 gene parts, we got mutations in the cis-regulatory region to cause problems with MYC, and on the other hand, we have mutations in the coding region to cause problems with RAS. Yes?
So does this cause a frame shift mutation in exon 12?
No, because they're showing here in yellow the coding region. And this is a picture of what we haven't mentioned before, and that is something about this RNA processing occurs. So this up here is showing that original transcript that's made from the DNA that now we're going to call the primary transcript. And then down here, this is showing what leaves the nucleus. And in between, there's this RNA processing. And so there's a mutation that causes a problem in RNA processing. And if you look, here's this, and just to add another term, so we have the primary transcript, that's a good name, first transcript, and then what leaves the nucleus is called the mature transcript, like that, and in between you have this RNA processing, and you can see here's the mature transcript, and there's the coding region in yellow. So what it's showing is that the coding region is not all contiguous in the primary transcript or the gene. That's fine. Part of this RNA processing involves something called RNA splicing. And that's a chapter we skipped over, chapter 6, right? That's fine. Just be aware that this is a mutation in the pathway of gene expression that affects not transcription, not the protein, but that processing pathway. So we have kind of a double whammy with this mutation story. First, it's a seemingly boring protein that is the gene that gets mutated. Lamin, whatever, you know? It's just a structural component of the nucleus. Like that? Seems like it, right? But it can't be. There must be something really fascinating about it. Then secondly, the type of mutation is a different one. It's not the coding region. It's not about transcription regulation. It's not the protein. It's not about the protein product and post-translational activity. It's about this RNA processing. So we get kind of two for the price of one. And we don't need to dive into the details of that because we didn't talk about RNA splicing in our course, but I can answer questions about it. Just realize that, right? The book is choosing this example with intention, right? Where probably everybody's heard of this rapid aging genetic disease, and then it's a surprise. Here's the surprise. It's twofold. One, it's in what seems like a boring structural gene, and two, it's in an unusual, curious part of the gene that affects that RNA processing, that RNA splicing that needs to occur for the gene to be functional and expressed. Yep? Yeah, and the book dives into us a little bit, and you know, it's a thing, right, that molecular biologists hate because molecular biologists is like, this is not, you know, it's not nuts and bolts. We just want to focus on nuts and bolts. You bring in a human interest story like progeria or cancer, and a molecular biologist is like, all right, let's keep it simple and keep it focused on nuts and bolts, you know? Like when I go to the car mechanic right over there, and I want you all to go to Woodford Street Garage because Stevie and John are really nice. They don't want to hear what I'm going to use the car for, you know? They're really good mechanics. And I'm like, we're going to Florida. And they're just like, your car doesn't have any oil, you know, like that, right? So it's part of that too. So we just kind of roll on with fascination. So again, it's part of this story, right, where we're trying to add some of that to our course at the end without confusing you, just adding to the basic framework and building on it. There's another image, so back to that nuclear pore, because we're really interested in the gate, yeah? And so, again, another different drawing of the structural component of the gate, because it has these little fingers out in the pore. And you can see is that, like, that looks like a car wash, and you're like, well, what are you gonna do about it? You know, you're like, you gotta get through this, you know? And you're like, well, I need help, right? Because the nuclear localization signal is simply a tag for another protein, a set of proteins that can bind to those and say, let's go through this car wash right now. Yes?
Can you go back to the photo before?
Maybe,
Is
that one?
Are all of those?
Yeah.
Or so there's multiple of them, like.
Yeah, I think that they've definitely, they're in the cell, they're scattered.
Okay.
There are many of them, but they're scattered. And I would say that they've done something here to kind of try to cluster them to make a, you know, a clearer image like that. I assume. Yep. Okay. So there's a little bit more of that nuclear localization signal that I talked about before. So it's typically just a series of positively charged amino acid side chains like that. So pretty hydrophobic and hydrophobic with a charge, right? It turns out it's just a binding site for a different set of proteins. Yes.
Aren't charged amino acids hydrophilic?
Yes, you're right. Sorry, did I say hydrophobic? Yes, hydrophilic. Yep. And and it's going to be a binding site, and it's a binding site, and we're faced with another situation where it's a binding site for a whole class of proteins. And our book has a list of them, right? Introduces them, and even if you want to get all the points on the final, we're just going to learn one name of one of them. So we dealt with this before, and I didn't do a good job. So this time I'll do a good job, and I'll show you a table that shows examples of that. And we're just going to learn the name of one, and it's got a great name. It's called important. And the function of important is to bind to a nuclear localization signal and help the protein get into the nucleus. So it's importing, and it's important.
What's the name of the signal you said?
The second one I said,
No, you said it binds to the something signal.
the nuclear localization signal, the NLS, right? So, you need an NLS to get into the nucleus. What's it good for? A binding site for a protein like important. And some of these other proteins might come in, but whereas for getting into the nucleus, we'll focus on important. That'll be great. And so here's just a little image of it. And so there it is. Here's the nucleus below. Here's the cytoplasm. Here's any of the proteins we listed that have to have a nuclear localization signal to get in a nucleus. So transcription factors, histones, DNA polymerase, and so forth, right? So they're going to bind to a protein like important, and important will get them into the nucleus, and then it lets go. So it's an importing protein, and it's important. Yeah, right? So those biochemists came up with a good name, right? And they probably walked right to the great lost bear when somebody said it. Okay, so then we want to have one more piece of this, and that is that this important protein is cycling in and out of the nucleus. So it's just an Uber driver to get things into the nucleus, right? It's waiting for a rider out in the cytoplasm, binds to one that's got a nuclear localization signal, and it brings it into the nucleus. And it's just cycling. And here's the only time we'll talk about this, probably in our class. So our class, of course, is almost over. But notice that this is a really complicated cycling pathway, and it includes GTP. So it turns out that this is an energy-dependent cycling process, and you can think of it like paying an Uber driver. And it's curious in the cycling process, right? Its job is to get something in. In the cycling process, where you have to pay the driver is to let the driver go back for another rider. So this brings up the next thing, and that is, not only do you have to import things, but you also have to export things. And so we have to export important after it drops off its ride. Yeah? By having it energy dependent, that makes it like a switch, like a teeter-totter, rocks back and forth. And that keeps it going, and that drives the energetics of this whole process. I think we've talked a little bit before about how, you can have a light switch, but you don't want it to be just really floppy. You want it to have to be sort of a switch that requires some force to switch. And so we are used to thinking about metabolism and ATP or GTP as a thing that's involved in, in synthesizing new molecules. But there is energy flow in the cell, and that's really often energy dependent because it creates a switchable directionality to it and a regulation of that overall cycling process. Yep, Alex.
I might have missed this, but is that a protein?
Yep, important is a protein.
Does it also have NLS?
That's a good question. I don't know. Does important have an NLS itself? I don't know. I kind of doubt it because I think it's interacting with the car washing and the nuclear pore using a different mechanism. Yeah. So probably not, but I don't know. Other questions people have?
I think it's kind of obvious, but that cargo protein would be like a transcription factor.
Yes, the cargo, does it call it that here?
Yeah.
Yeah, the cargo is any of these proteins we talked about that has to get into the nucleus. Yep.
And in this picture, I'm sure there's probably many, but in this picture, is that just one that we're seeing go down and then make all these different like interactions? Like that's one important? Or is that many kind of all doing it at the same time?
So, I didn't look at this before class, I have to think about this. So what we want to just, this shows the whole cycle, and we want to just absorb the important part first, right? The importing, okay? Because there's export too, okay? So let's come back to this to look at that, how it's getting out of the nucleus. But for now, just realize that there's a cycle going on. It's the same molecule of important that's going in and out and in and out, right? Like that. And to do the cycle, it requires inside the nucleus this hydrolysis of GTP. And then we'll come back to that, Okay, so let's just stick with the important a little bit and just show this. Let's see, make sure we get this. So, here, as I already mentioned, the nuclear pore it's a gate. It lets small molecules go in freely, but these larger molecules, there's going to be regulation. And you have to have, if you're a protein, a nuclear localization signal. And so that goes back to Matt's question, which is a good one to think about, and that is, well, the virus is going to have to get into the nucleus. If it's a retrovirus, that DNA has to get in. And in general, I'm pretty sure that DNA and RNA always need to be bound to a protein, and the protein has a nuclear localization signal. So DNA or RNA gets in also indirectly using a nuclear localization signal. And that's where some of the other importing proteins come into the story. We're just going to learn about important. Important is really focused on proteins that have a nuclear localization signal and just bringing those in. There are other proteins that have nuclear localization signals that have different additional cargo, and they use some of these different help to get in, different importing. And we just don't need to learn the name of that whole class of proteins or the other proteins. Then the next, yep.
Is important itself a class, or is that a family, or is that a specific protein?
It's definitely, for us, it's a specific protein. It's a specific protein, and then the more that would be speculation. Is the family? I don't know. Yep. But it's not I would say it's probably not a class. And for us, definitely want to think of it as a specific protein. Okay. So then the next thing we want to bring up is that there is a pathway out of the nucleus, and it's also regulated. And one protein we're going to learn that is for exporting is called exportin. So that's satisfying too. And it also has this cycling process. It's also energy dependent to make that go. And I guess that I never thought about an Uber driver before, but I guess that's kind of accurate, right? The nucleus is like a show in downtown Portland, and you're like, we got to get a lot of different people from other places, in town to the show. So we get an Uber driver, and they're just cycling back and forth, driving you off and getting paid. That's the energy value of it. And so you got to get out of the nucleus like that. And let's see, I don't know if I think the book might have dropped this slide. In previous editions, it would show that that's where it likes to show that RNA needs to be bound to proteins all the time. And when the RNA leaves the nucleus, it needs to leave the nucleus using something like exportin. And the exportin is not recognizing the RNA, it's recognizing proteins that are bound to it. Yeah, and I guess our book dropped that figure because it just shows RNA, it shows proteins, it shows the RNA leaving. But that's significant, right? Because we talked about how when RNA leaves the nucleus, if it's messenger RNA, it's called mature RNA when it leaves the nucleus, right? So it's regulated, right? The RNA gets this RNA processing and splicing to go from a primary transcript to a mature transcript, and then it's allowed to leave the nucleus. Who's in charge of that? Exportin and the proteins that are bound to the RNA. So there's that, okay? So then then the next thing to say, and then we'll loop back to that other, is that now we've established, there are these two stories. And then the GTP energy story has a protein that is bound, that's a G protein. And we've learned about G protein. G proteins are bound to either GTP or GDP. And so that's what's happening here, is that a G protein called RAN is regulating the process of converting either exportin or importin into a exit pathway. And I think you see it here, you can see it's just like that. But I think it's a little satisfying myself to look at it with regard to the important, because important is we're just trying to get cargo in. And then you're like, okay, now important drops it off. And important's got to get out of there. So there's got to be a directionality to it. And you don't want it to leave until it lets go of its cargo. right? So the Uber driver should stop in downtown Portland, let the people out before they go out to get more people, right? There's got to be some regulation of the cycle, and it has to include dropping off the cargo and not just hauling it back out with you again. You know, you got to drop it off. So that's part of this cycle and the concept of having an energy-dependent directionality like that. And after it drops off cargo, important gets bound by this RAN, and the RAN hydrolyzes GTP and kicks it out the path out of the nucleus like that. Yep.
So you said it was a G protein. Is it the same as the G protein coupled receptors with like the drug
No.
protein? Okay.
Nope, it's not like RAS, and it's not like the trimeric G protein. It's just a different G protein. And why is it called a G protein? And it's just because it uses GTP and GDP in that active-inactive strategy. Yep. Yeah.
And then so it has inherent GTP-ACE activity.
I don't know. I don't know anything about this RAN like that. Does it just spontaneously hydrolyze? And I don't want to speculate, but you can. Do you think it does?
Be a good place to start asking.
Well, see, so it's supposed to help important get back out. So it should probably hang out active until it binds it important and helps it out, and then it should hydrolyze it, right? So I suspect it might not be spontaneous. It might be regulated to say, get the job done, drop the people off, get paid, and then go get more. And
anything that's not regulated, we've seen in this class time and time again, leads to chaos.
yeah, there it is, tattoo. Yeah, anything that's not regulated leads to chaos. Yeah. Okay, great. Speaking of which, I just remembered, and I'll make a slide of it, the biology majors are going to play the chemistry majors in a kickball game on the last day of classes.
Where? They took our field.
Yeah, so we'll just take over that construction site. That's not a problem. But you'll see signs up, and Meg Hausman is always motivating this.
Our grad students a lot.
But yeah, last year, just a reason to get out, I think last year the biology department majors won maybe 23-1. So yeah, you don't have to participate. You can just come out and chant and shout and get some, like that. Yep. Okay, so just a couple of highlights that we want to get out of this, and that is, there's a gate. So there's the potential for regulation, and there's a gate leading into the nucleus. Nuclear localization signal is required. We've got examples from our course where that's regulated. FOXO and heat-shock transcription factor would be good examples of it being regulated going in. Then stuff is regulated going out, and we haven't seen any specific example of that, except for this one that just came up. Post-transcriptional regulation includes RNA processing, and that has to happen inside the nucleus. So we won't let the RNA out until that process has been finished. Good. Okay? Questions about that? And we want to drive through this, right? Again, there are more examples and stuff like that. And I said at the end of the chapter, we'll pick up this example of how to assemble a ribosome, because that'll bring us back to this export-import process and add a little bit to it. Yep? Questions about that? Why don't we stop for concept chat, and then we'll talk some more. So we argue about, who knows how many weeks we have left in this semester of classes? 2 weeks of classes and then finals week. Yep. So we have our final 2 weeks from today.
No, 3. So we've, including this week, 3 weeks, I think.
Oh, man, are you guys just want to stay in school? I want out.
We can do it a week early.
Yeah. Let's make a class plan, right? And just be like, we're running on our own schedule here.
One of my seminar classes, we decided to stay late one day, so we didn't have to have the last day for finishing
Yep.
presentations.
Yep, that's tough. All right, see if you can meet somebody else. Is there somebody else that you haven't met? Like Lydia. Has anybody not met Lydia? Or Anna, or Ben, or Isabella, right? The people that sit in the corners. There we go. We got some movement.
No one likes this.
Okay,
Fine.
well, on the board.
Okay. Okay, so we're working on the nucleus now, and there's the nuclear pores. They're not sesame, like they're not just one.
Yeah, find something else.
The protein. Yes. So they have these pores that are regulated. They're lipids.
Yes.
And they use energy. Ribosomes are always on the outside of the nucleus. Outside of the protein. I am confused by the NLS as still. I know that it's a nuclear localization signal. Is that so? That binds to transcription factor, let's say, is what you want to get in, and then the important will bind to that, which will bring it in.
Important bind to and
Oh, man.
From my understanding, like looking at the picture, I thought it looked like, I don't know, I got to run that check here, but I thought it looked like if you wanted to bring in bound to the NLS, and then important bound to that, and then they would, but I don't know. That's like my understanding. But then once they got in, then you have to have, and you don't really need to get in, I believe. I believe you can on your way out. Right? Yeah, I understand. So it's, I think there's like, that's a good question. I don't know. I don't want to speculate. I'm assuming they're both in and out. Yeah, you should.
Good question.
What?
Change.
Are the pores specific to in and out? Like, is there in pores and out pores? Or are they?
Both
Just the
ways.
proteins are specific. Okay.
Yeah, it's my understanding, yep.
So, okay, one more question.
Yep.
So if I wanted to get transcription factor in, does the NLS, is that right, the NSL, does that bind and then the important binds and then it goes in?
Well, the NLS is part of the transcription factor protein. Yeah,
Oh,
it's
it's
just
part
part
of the protein.
of the transcription factor protein. Yeah.
So then, and you don't use HTTP to go in, so like you don't pay to get into your Uber, you pay once you get there. Perfect, okay.
Perfect, yep. Okay, so let's see if Mallory, what's one thing your group talked about you thought was important today?
We talked about import and export, as well as like how it works with NLS to like regulate everything that goes in and out of this.
Right, and the NLS is working on the import side. And I don't know exactly what the tag is that, or tags might be for the export. Like, what is export and recognizing? And I don't know. Sorry, limited knowledge. Oh, and what's another thing your group talked about?
Leaving the nucleus, mature transcript.
Yeah, right. So that's part of the story of gene expression is that the DNA is in the nucleus. That's where the genes are. They're going to need to be transcribed to be expressed. They're going to get transcribed as RNA. The RNA is going to need to get out of the nucleus, and that's a regulated process because there's a post-transcriptional regulation of gene expression that takes place in the processing of RNA, including this RNA splicing and other events that have to happen inside the nucleus. And then we have mature RNA that gets specifically exported. And Brian, what's another thing that your group talked about?
Progeria, just like interesting how just like that mutation in the nuclear lamin can
Yeah.
cause.
Yeah, I mean, just just to imagine, right, that those kids were suffering from that disease, and they know it's a inherited disease, right, like that, and then they finally find the gene, and it's a single gene, and they're like, what? A mutation in one gene can cause rapid aging, and they're like, well, we're interested in that gene, and then they find the function of the gene it's to code for this lamin protein, and they're just like, yeah. And I don't know, I mean, you should go home and Google it to see what the status is, if somebody's got an understanding of like kind of the biochemical mechanism, what's going on with that disease, like, what's kind of defective in the, the cell biology of the cells to cause them to age rapidly like that. Yep. Okay, and so we'll keep talking. Do people have questions about anything at all? So let's look at this figure in the book. Here, this figure in the book wants to do what we already did, which is to pick an example of a transcription factor and show how its entry into the nucleus is regulated. And this is a transcription factor that came up in chapter 16. Toward the end of the chapter, we talked about, there's beta-catenin, there's NOSH, there's SMADs. This is one we just didn't talk about, and we don't need to talk about it now. because we have really good examples of the regulation of importing transcription factors in a nucleus. This is just a different example, a different story. Our examples are FOXO, that's a classic one, super satisfying, and heat check transcription factor. The transcription factors have this NLS, it's just part of the protein. And it might be blocked. It might be blocked by just the way the HXF is folded. It might be blocked by 1433. And that's what's happening in this. And again, you don't need to understand this one. But there's another protein, a subunit of this protein that's blocking it, and the subunit has to get destroyed. You know, like that. It's a super famous protein. It's a really important in what Sam has been telling us about. He's going to tell us one more time about. So it's really important in the activation of our immune system. Inside the immune system cells, when they make genomic responses, this transcription factor plays a big role. So that's just an example where we don't need to dive into it on our course, but you could figure it out, you know, like that. Like maybe you picked a receptor for your term paper, and the receptor activates this transcription factor, and you're like, okay, I can figure this out, right? So that's, you know, something we try to talk about now later in our course, is that we make this framework and you're like, okay, that's that, right? So that's that picture. And then then we just want to talk about this FOXO a little bit more, because we've talked about FOXO and we haven't really, we probably have learned that word 1433. You know, it's a pretty lame name for a protein. And but it turns out that 1433 is a family of proteins that are really, really interesting proteins. And people have been studying for a long time. So now we just want to build up that slide that we started our course with to say, what is 1433? And it's got to do with this cellular localization. So it can keep things out of the nucleus. That's a thing it can do. And so we're just going to look at that a little bit more and just add a little bit to that, because there's a lot more that could be added. So let's just look at this. So 1433 proteins regulate cell cycle progression by interacting and modulating with the activity of CDC25 phosphatase.
Oh, we know about that.
Yeah. So maybe I'll just scroll off of this and we'll read a little bit more about it. But just remember, during our study of the cell cycle, we really wanted to study the G1 to S transition start, and we did some. That's where we learned about p53 and retinoblastoma and small inhibitory proteins and so forth, like that. But we also wanted to learn a little bit about the cell cycle at first by going over to the other side of it and talking about the G2 to M transition. And we said we'll learn about CDK1, cyclin B. When they're active, that's called MPF. And when they're active, a cell is in M phase. And at the G2 to M transition, there's a DNA checkpoint. And if there's DNA damage, we don't want to go to M phase. We want to repair the DNA first. And the protein that's important is this CDC25 phosphatase. And it's going to remove inhibitory phosphates from CDK1. And that's what's got the brakes on, remember? And students get confused about that because we also learned over at START about other topics about a DNA damage checkpoint. But, so it's just important to remember that. And it was that simple. So the DNA damage occurs, and check 1 phosphorylates CDC25, so it can't do its job, right? So it turns out the way that gets blocked is 1433 binds to the phosphorylated form of CDC25 and just holds it in the cytoplasm in the same way it holds FOXO. Because almost all of the CDK1 protein is inside the nucleus. You know? So there's lots of different ways to inactivate a protein by phosphorylation. Turns out one way is to just phosphorylate it and just have 1433 hold on to it, and people call that, they say sequesters it in the cytoplasm. It just doesn't have access to removing the inhibitory phosphates like that. And so we could replace FOXO here with CDC25. We would put CDK1 in the nucleus, and they just can't get together. So, you know, that's just like, poosh, right? Because the first time I talked about that in this course, we'd been talking about FOXO, and we had only mentioned 14.3.3 once at the start of the semester. And at this point in the semester, I asked students if they knew what might bind to phosphorylated CDC25, and a student named Dan said 14.3.3. I gave him an A. So, yes?
Can you tell us where P16 acts on the cell cycle?
Where does P16 act on its cell cycle? Yes. So all of this, it's a small inhibitory protein, and all the small inhibitory proteins act at start. Yep. Oh yeah, that was a question on the exam. Okay, so here's an add-on to introduce this, because the 14.3.3 story works on FOXO and the regulation of FOXO activity, and it works on the CDC25, and then a bunch of other stuff. So that when we look, it says 14.3.3 proteins regulate cell cycle progression by interacting with and modulating the activity of CDC25 phosphatases. Right? When they're phosphorylated, they're inactive. Check one phosphorylates them, phosphorylates CDC25, and then 14.3.3 grabs it and says, hold on, buddy. Yep. CDC25 phosphatase like CDC25 play a crucial role in activating cyclin-dependent kinases, CDK1, which are essential for cell cycle progression. Like that. Blah, blah, blah. I just want to point out, 14.3.3 binding can inhibit CDC25 activity by preventing its translocation to the nucleus, where it would activate CDKs. And in our case, specifically, we're interested in that CDK1. Okay? Sweet. Right? So this is just a question, you know, something about like, where is CDK? Is CDK1 in the nucleus? Yes. CDK1, also known as CDC2. Forget about that. We talked about that. It's just a distraction by old-time geneticists. So, yeah, CDK1 is present in the nucleus, particularly during mitosis. So this just shows how CDC25, when there is DNA damage, gets phosphorylated and gets sequestered in the cytoplasm. Just a little visual to what we just said. Yep. Okay. Remember we talked in our course early in the semester about FOXO, and we said, you know, you could build a transgene where you add GFP to the coding region of FOXO, and then you could see the cellular localization of that protein, right? So there are lots of experiments like that, you know, where you're studying 14.3.3, and we're like, where are the proteins at? You know, where can they go? Yep. So here's just a little thing, because there's quite a bit of complexity around regulating CDC25. But drawn here, we see 14.3.3 and this idea of sequestering. It's just a normal English word, which means we're going to sequester you, we're going to keep you in a certain location. So they're going to sequester the protein in the cytoplasm. It can't do its job because its target, CDK1, is in the nucleus. So that just goes back to, we're adding to that FOXO example. Totally different stories, but they have in common this 14.3.3 gene family. And so it's 14.3.3 plays a role in stress. It's different than the stress that we talked about with heat shock. And it has to do with the stress that like FOXO is regulating. So FOXO is regulating a plan to slow down aging if the environment is harsh. So if there are like limited nutrients and the environmental conditions are poor, organism should slow down its reproductive maturation, slow down its aging, and cross their fingers for better conditions. Yeah?
Is that what causes like late puberty in gymnasts and dancers?
Is that what causes late puberty in gymnasts and dancers? I don't know. But here's something that causes early puberty, and that is excessive calories. So, you know, it's kind of famous in modern society that puberty is beginning at younger and younger ages. And people worry, is that a pollutant? What's going on with that? And the answer is nope. If conditions are good, literally an animal, like us, should mature, reproduce, and die. Just put all of its energy into reproduction and die. Yep. So yeah, just lots of calories, and and in humans it's not dramatic, right? It's earlier, but still a lot of different range and ages of people. In some species, it's really dramatic. It's just really locked into the environmental conditions, and if the conditions are poor, they just slow down. They even go into a kind of hibernation and just wait it out, hoping, crossing their fingers. It could even be that they're crowded. It's a crowded environment with a lot of competitors of the same species, and they're just like, we're just going to wait. We're going to try to hope for better conditions. On the other hand, if nutrients and conditions are positive, reproduce, kick the bucket. Yep. So it turns out this 1433 family has lots of different roles. The cell cycle is one of them, right? Is it involved in stimulating the cell cycle? Not in our example. It's involved in slowing it down, right? Which is all about, let's hold on. So if there's stress, let's slow down. Yeah, Alex.
Is that what nematodes do when they enter the...
Yes. So there's a little molecular model organism called a nematode. It's like a fruit fly, and they're really famous for they can just stop everything and extend their lifespan and wait for better conditions. Yep. So just a little bit about these 1433, right? Because we started our course and we're like, that FOXO is certainly an interesting protein because when it gets to do its job, it activates a whole bunch of genes that slow down aging. So this 1433 is a fascinating family of proteins that are a family of structurally related similar phospho-binding proteins that regulate essentially every major cellular function, right? And this is a thing where you go like, I'm going to do some summer reading, maybe it'll be about 1433, right? Maybe go to the beach and just be like, nerding out here today because it's fascinating. And we have that framework to start diving into it. And so decades of research on 1433s have revealed a remarkable network of interacting proteins that demonstrate how 1433s integrate and control multiple signaling pathways. In particular, these interactions place 1433 at the center of the signaling hub that governs critical processes such as cancer, including apoptosis, cell cycle progression, autophagy, glucose metabolism, and cell motility. Historically, the majority of 1433 interactions have been identified and studied under nutrient replete or abundant conditions, and they've important roles for nutrient-driven interactions. However, this underestimates the reach of 1433s. The loss of nutrients, the loss of growth factors, other environmental factors can really show us what the role of this player is. Then I just wanted to highlight a couple of sentences down below. So here's one, and it is this dynamic nature of the 1433, and they call it an interactome. And they're just saying, let's talk about everything that this family of proteins can ever interact with. Let's know it all. Let's study it all. And so they will learn in our last week of class, depending on if we have 3 weeks, we'll talk, we won't read about it, but we'll talk a little bit about this idea of like, let's learn about all of it at once. And they will add an like genomics and things like that. And so they're saying that, let's learn about it all. And then they mentioned this instrument called a mass spectrometer. And it's a way of just looking at all the proteins and the status of all the proteins. So we got to stop there. We'll look at the next little section of 1433s, and then we'll move on like that next time, okay? And I'm going to try to have your exams graded. I apologize, because I know it's anxiety provoking until you get it back, and I'm sorry about that.